Let's talk about mitosis, which is the process of one cell becoming two exact daughter cells. Before we go into mitosis, a cell has to go through interphase. An interphase is when the cell actually increases in size and all the contents duplicate in preparation to become two cells. Then we go through mitosis, which is what we're going to talk about in this discussion, and this is followed by cytokinesis, which is when the one cell actually physically pinches off and becomes two cells. In this process, cytokinesis overlaps with the last two stages of mitosis, but we'll talk about that more in detail later in the discussion. First, some definitions, starting with chromosome. Chromosome, we all know that's the DNA and the associated proteins that are going to make up the genetic material of a cell. And when it's going through interphase, when the chromosome is duplicating, it's going to be loose like this, so all the enzymes can have access to it that are necessary for the duplication. But you'll hear me refer later on that during mitosis, the chromosome is going to condense, so we're going to show that as more of a solid blue structure here. And since it's already duplicated, it's going to be two. And when it's duplicated, we refer to the duplicated chromosomes as sister chromatids. Next, we have the nucleus, which is going to contain the chromosome, and it has a nuclear envelope, which is a double membrane. So we're going to show that as these two green rings, and you can see that the sister chromatids are within the nucleus. Next, we have the cytoplasm, which is the contents of the cell that are outside the nucleus but still within the plasma membrane. And we're going to show that as this purple shade here. So you can see that it's outside the nuclear envelope but still within the plasma membrane. Next, we have the mitotic spindle. Now, this can be a confusing concept, but it's really pretty basic if you get the definitions down. The mitotic spindle is nothing more than the microtubules during mitosis. So whenever it's a single cell and you have the microtubules, whenever they've duplicated and they're going through mitosis, we refer to the microtubules as mitotic spindles. In the same sense, the spindle poles are nothing more than the centrosomes during mitosis. So when you have a single cell, they're centrosomes, but whenever they've duplicated and they're going through mitosis, we refer to them as spindle poles. So to make this a little clearer, we have a picture here where you can see that there's just a single centrosome and single tubules associated with that centrosome. But whenever that centrosome and microtubules, whenever they duplicate during interphase and they're starting to go through mitosis, we refer to them as spindle poles and mitotic spindles. So the centrosomes become spindle poles and the microtubules become the mitotic spindles. Next, we have the centromere, and the centromere is going to be the structure that holds the sister chromatids together. And this is also going to be where the kinetochore is located. And the kinetochore is where those microtubules of this mitotic spindle are going to attach to the centromere during mitosis. And we'll talk about that later in detail also. And the centromere we're going to refer to as this pink dot. So you can see it here, it's holding the sister chromatids together. So the centromere holds the sister chromatids together. Now let's go on to the 5 stages of mitosis. So the first stage is prophase, followed by prometaphase, followed by metaphase, anaphase, and telophase. So there are 5 stages of mitosis. The first stage, prophase. This is where those chromosomes are going to condense. So before an interphase, when they were loose and squiggly so the enzymes would have access to them, they've now completed duplication and they're condensing into solid forms, which will make it easier for them to divide. So the chromosomes have condensed. And also those centrosomes, which were replicated in interphase, are going to start to move apart and become the mitotic spindles. So the mitotic spindles are beginning to form. Okay, so the centrosomes are moving apart and the microtubules associated with them are moving apart and the mitotic spindles are forming. Then we transition into prometaphase. And in prometaphase, the nuclear envelope is going to break down. And when the nuclear envelope breaks down, it's going to allow those microtubules of the spindle to have access to the kinetochores on the centromere, and those microtubules are going to attach to those kinetochores. So in prometaphase, the nuclear envelope breaks down. And the way I remember this is I take the first 4 letters of prometaphase, which is prom, and this probably isn't very PC, but if you've ever heard the phrase off like a prom dress, I remember prometaphase, the nuclear envelope is off like a prom dress. So in prometaphase, the nuclear envelope is breaking down and the microtubules are attaching to the kinetochores. Then we transition into metaphase. And in metaphase, the chromosomes are meeting in the middle, so they're lining up on the equator of that cell. And how I remember what happens in this stage is that met, the first three letters of metaphase, met is kind of like meat, and the chromosomes are meeting in the middle during metaphase. Then we transition into anaphase. And in anaphase, the microtubules of the spindles are shortening. And as they shorten, they're going to pull apart the sister chromatids to their respective ends. So the microtubules are shortening, and as those sister chromatids are pulled apart, they're becoming their separate daughter chromosomes. So the sister chromatids are pulled apart and they become their daughter chromosomes. And how I remember what happens in this phase is Anna, the first three letters of anaphase, and daughter chromosomes. I think Anna is the name of the daughter chromosomes are forming. And then we transition into the final phase of mitosis, which is telophase. And in telophase, the daughter chromosomes have arrived at the spindle poles. So those microtubules have shortened where the daughter chromosomes have arrived at the spindle poles, and the nuclear envelope starts to reassemble in preparation to become two separate nuclei. So in telophase, the daughter chromosomes have arrived at the spindle poles, and the nuclear envelope starts to reassemble. Also, you have a contractile ring that begins to form around the middle of the cell, and this contractile ring is going to contract and contract until finally the one cell is going to pinch off and become two separate cells, and this is cytokinesis. But going back to telophase, so the chromosomes have reached their poles, the nuclear envelope reassembles, and the contractile ring forms. Now remember what happens in this phase is telo, it's kind of like telephone, telophase, telephone. And I think what's happening is because all the structures have reached the poles and those daughter chromosomes, they're so far apart that they're not able to talk to each other anymore, so they have to telephone each other in order to talk to each other. So in telophase, the daughter chromosomes have reached the poles and they have to telephone each other. That's how I remember what's going on. And then following mitosis, we've completed the fifth stage. Then we complete cytokinesis, which is whenever that contractile ring is actually going to pinch off and make two separate daughter cells. And it's important to see that cytokinesis isn't completely a separate stage all on its own. It overlaps with stage 4 and 5. It overlaps with anaphase and telophase. So you can see in anaphase and telophase that the structures have already kind of started to split. Those sister chromatids have split and become daughter chromosomes, and they've gone to their respective poles. So they've already started that process of becoming two separate cells. So cytokinesis overlaps with the last two stages of mitosis, anaphase and telophase. So as a quick overview, mitosis is preceded by interphase, where the cell is going to enlarge and all the contents are going to duplicate. Then you go through prophase, then we go through prometaphase, metaphase, anaphase, and telophase. And then we have the completion of cytokinesis, which is going to overlap with anaphase and telophase. So this is mitosis.